Hello, Bio 121. Welcome to Chapter 15, how we actually go from genes to making proteins. And at this point in time inside the textbook, what we're doing inside this class, of course, is we're layering on to the central dogma of biology. And the central dogma of biology is now that we're going to go from something we're going to call DNA. It will be transcribed into an intermediate called RNA, right? And then that RNA will, depending on what type of organism we're looking at, will bind to ribosomes, and we will now begin to make proteins. And when we say this is the central dogma of biology, what we mean is, if you think about actually the way this worked, well, we identified things like proteins first, and then it would take quite a while for us to actually identify what DNA was, and then it would take years again to figure out that there's this intermediate. It's called RNA. And at the end of the day, when we think about how Mother Nature probably put all of this together, probably all started with RNA, okay? And then RNA would lead to some proteinaceous state. And over the course of evolution, 500 million years or so, we want to make sure that you get this idea that the more complexity required the creation of specific stable sets of genes that are housed in these linear structures that we call DNA. And we'll see how that actually works. Okay? So, now, one thing you're also going to notice is that a lot of the content begins to sort of become repetitive here. And what I want you to do is be able to notice there's a difference when we're talking about prokaryote and eukaryotic cells. Remember, prokaryotic cells are those cells with no nuclear envelope, and they have essentially instantaneous translation mechanisms that are going to happen. Before we even get there, we need to think about sort of the general idea of how it is you're going to make a protein. Remember, back inside chapters 1 through 5, we introduced this idea of how it is we're going to use amino acids that are brought to a piece of information. That's a piece of information now, RNA. And those amino acids with a ribosome, okay, are going to build piece by piece, essentially the proteins that are going to go from a primary structure into a secondary structure and into a tertiary structure for them to be functional, right? And of course, some proteins require complexes, and they, of course, would be quaternary structures of tertiary structured materials. So all of this hopefully is coming back into your memory. But let's begin with, what are those amino acids and how are they grouped? Okay, because this is a human convention that bears out biological activity. So we start with, here are groupings of amino acids. So protein amino acids in both sets here. You can see that there are polar, non-polar, aliphatic, or uncharged groups here. You can actually have positively, negatively, and non-polar aromatic groups. And each of them have, sort of similar to what I was saying way back when in chapters 1 through 5, they each have a distinct characteristic. And that characteristic, you might recall, is built off of this amino terminal group, our carboxyl group here, and our functional group, the R group here. That R group is represented through each one of these modifications to that backbone. And one thing I want you to pay attention to here is, let's pay attention first to this amino acid, methionine. So it's nonpolar, okay? And what we want to make sure we see is that every time we actually build a protein, we're going to start with a methionine. That ATG sequence is going to allow us to be part of the signal that will actually cause proteins to be made. Now, we weren't around when Mother Nature came up with the why and the how and all of this, but what we can do is we can step back and go, well, it is a repeating characteristic, and we want to make sure we pay attention to that repeating characteristic. Okay. So, like I said at the beginning of all of this, there's a central dogma in biology where DNA is transcribed into mRNA. And you're going to see that the farther we go along, we finally get into chapter 16, even mRNA itself can be broken down into, well, how do we actually make mRNA? Because let me give you a little bit of an insight here. Well, DNA, from the previous chapter, we had to use topoisomerases, we had to use helicases, we had to use polymerases to transcribe a copy of the DNA so we could make a semi-conservative model. Well, we're not doing that here. Instead, we're using a series of RNA polymerases. What's going on here? Polymerase, so I made that mistake again. Let's hit pause. What we're going to do is we're going to work with this material we're going to call RNA polymerase. Okay, now that I fixed that little mistake, RNA. I'm just going to put Pol here, P-O-L, and you'll see why later. Okay, so RNA polymerase is now going to help us execute this central dogma. And this is one of the things you need to pay attention to. Are we talking about transcription and translation, so therefore we use an RNA polymerase? Are we talking about that semi-conservative model of replication of DNA during the S phase of a normal cell cycle? So you need to be a little bit discriminating when you're thinking about what's happening here. And this gets to, when we talk about, especially in bioinformatics, what populations of information are we actually looking at? You need to know what created the population of information you want to assess. So when we're thinking about how it is DNA becomes RNA, notice we're talking about a specific type of RNA, messenger RNA, and that messenger RNA is going to have to be pooled in some way or modified in some way, and those will be translated into proteins. So how does this happen? A very rough sketch of what is going to occur here. This is pretty much what I've been telling you all along. An RNA polymerase will come in and you're going to make some structure that has both exons and introns. And exons are the coding sequence that will become a protein, and introns are those sequences that we will cut out. They have regulatory responsibilities. They're not unimportant pieces of information. And when we actually have to make different isoforms of proteins, meaning proteins that may have slightly different shapes and sequences, but generally fall into the same responsibility of the same grouping of proteins, Mother Nature needs to know which one of these to actually modify. So we'll take this thing that's actually called a pre-mRNA at this point in time. We'll modify it, we'll splice it, we'll make some changes. Take a look at this cartoon here, take a look at that cartoon there. Pretend you're back in grade school and say, well, we got purple dots here, and we've got polyadenylation signals here, and you need to understand why Mother Nature is doing this. And this represents not just processing, but regulation. And once it's regulated properly or processed properly, we can now begin to actually translate it. And here's that ATG or AUG. And this is where our methionine is going to be, and that's where our initial methionine came from. You'll see there's a pattern at the ribosome. You remember ribosomes are these structures that are made inside the nucleolus of the cell. And this is a pretty good place for us to start thinking about how do we keep all of this information straight inside of our minds. And I'll see if I can help with that, but it just takes a fair bit of work. Okay. Now, I'm going to pause here because this takes us in a completely different angle, and I want to pick up with a new idea when we come back. But degeneracy of the genetic code means that we need to introduce to you how it is we can take 20 amino acids and make all of the proteins that we need to make for all living structures on this planet.